So did you people have any difficulty in the previous topics that we had discussed? Anything you people wanted to ask?
No, ma'am.
No, ma'am.
The different kinds of vectors were clear to you all?
Yes, ma'am.
Okay. Who can tell me what makes a vector ideal? How can you call a vector as an ideal vector or a perfect vector for recombinant DNA technology? Hmm? Say? No one? Okay. Anyway, what is the last thing you people have written in your notebooks? Yes, am I audible? Yes or no?
Yes, ma'am.
Tell me, what is the last thing you people have written?
Uh, um,
Let's just wait for 2 more minutes, then I'll start the class. Okay, everyone, switch on your videos. Everyone. So, until the previous class, we had discussed that what are the ways by which we can do RDT, meaning what are the ways by which we can do recombinant DNA technology? How can we create a transformant? How can we create a recombinant DNA? And we also saw how can we send it to the host, right? We know that ultimately, meaning after the whole entire process of this is done, what do? We send the recombinant DNA into the host so as to increase the number or so as to make the desired gene multiply. Students, are you not able to listen to me? I asked you to switch on your videos, everyone. I'll not repeat myself again and again. If I'm asking to switch on the video, then please do so. And no one will switch it off until I give you the time to take down the notes. No one. All right. So talking about the process. Now see, I told you that after the recombinant DNA has been created, it has to be sent to the host body or host cell so as to perform the multiplication, so as to make multiple copies of the gene of interest or the desired trait. But do you think it is like so easy to take the recombinant DNA and put it into the host cell? Do you think it is that easy? If I give you any cell and if I ask you to put anything into that cell, do you think, like, how will you do it? Will it be easy for you to do it, or will it be a little bit difficult?
It will be difficult, ma'am.
Yeah, it will be difficult. Why? There is a reason why it will be difficult, because the cell has its own properties, right? The cell has like a plasma membrane present. It has like a cell wall present in the case of plants and bacterial cells. So it's not very easy to break those barriers to send the gene of interest into it. So what do? We make the host cell competent. You people understand the meaning of competent? Hmm?
Suitable? Suitable,
Hmm?
like they make the host cell suitable.
Suitable. Suitable, or to be to make capable. Or like the word competent means to be capable to take up the gene. Meaning so that it will easily take up the gene that you're transferring into it. So that is what we mean by making a host cell competent. Now, there are multiple ways by which we have to treat the cell so as to make it competent. The first is treating it with calcium dipositive ion. Second is giving a heat shock to the cell. Then third is micro injection. Then there is gene gun and disarm pathogen. All of these are the different methods of making the host cell competent. See, the host cells, they have to be made competent to take up the DNA. Competent means to be able to take up the DNA, because we know that the DNA is a hydrophilic molecule. It is water loving. So it will not pass through the cell membrane very easily. So to make the DNA pass through the cell membrane easily, what do? We make it competent. Now, the first case is treating the host cell with calcium dipositive ion. Now, in this process, what we do, see, calcium ions, it has a great, you can say, connection with the cell membrane and its permeability. So, this calcium dipositive ion, it increases the efficacy with which DNA enters the bacterium through pores in its wall. See, this calcium ion treatment, it is done in the case of bacteria. Whenever you want to make a bacterial cell competent to take up the foreign DNA, whenever you want to make the bacterial cell competent, so we treat it with calcium dipositive ion. And because of it, the efficacy, meaning it becomes very efficient to take up the DNA now. Why? Because calcium dipositive ion creates pores in its wall. Pore means tiny holes in the walls of the bacteria. Now, incubation of the host with recombinant DNA on ice, giving heat shock at 42 degrees Celsius, and then again putting it back on the ice. So changing the temperature. First you put it on the ice so it becomes very, very cold. Then you give it a heat shock by suddenly taking the temperature to 42 degrees Celsius, and then again putting it back on the ice. So this causes the host cell to take up the recombinant DNA. Any doubts? The second one is micro injection. See, in this calcium treatment only, I have discussed this one and this one, heat shock. Putting it on ice, suddenly increasing the temperature to 42 degrees Celsius and then bringing it back on the ice. So you're changing the temperature very quickly, because of which the host cell will become competent. So first and the second, they are used for bacteria mostly, because the most you can say widely used organism for this RDT is bacteria itself. After that comes micro injection. So this is used in the case of animal cell, wherein with the help of injection, the recombinant DNA is directly injected into the nucleus part, because we know nucleus is the main control house of the cell. So the recombinant DNA is directly injected with the help of a syringe. It is injected into the nucleus of an animal cell. So microinjection is used for animal cells. Then comes the gene gun, or you can also call it as biolistics. Now the gene gun or the biolistics, it is used for plants, wherein what we do is that we take this thing. We take a gold or a tungsten particle and we coat it with the RNA, like recombinant DNA, rDNA. Okay, we coat the gold or the tungsten. See here, the gold or the tungsten acts like a bullet, just like how bullets are there in a gun. So same way, and then with the help of that, the cells are bombarded with high velocity microparticles. So a lot of microparticles of gold and tungsten. It doesn't mean it's like present in a big ball. No, tiny, very microparticles, and the cell gets bombarded. Bombarded means constantly thrown with gold and tungsten that is coated with recombinant DNA. So this is used for plant cells. And then the last one is the use of disarmed pathogen, meaning when the vectors, they are allowed to infect the cells, and by this the recombinant DNA is transferred into the host cell. Just like how I told you for retroviruses. If you remember, retroviruses, we disarm them, meaning we take their pathogenicity, and then because they have a property to divide a lot, so we use that property to transfer the recombinant DNA into the host cell. So that is what we do. Any doubts? Okay, so now you people can note it down. What is the last thing you people have written? Have you written shuttle vector?
Yes, ma'am. Completely. Without this.
Now you can note it down. Now for now, you can switch off your videos.
Ma'am.
Done, student?
Yes, ma'am.
Yes, ma'am.
Done, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Done, ma'am.
Okay, everyone's done? All right, now these are the ways by which you can make the host cell competent. Now we're going to dive into the actual process of how the recombinant DNA technology is done. So there are certain steps that must be followed in order to make or create a recombinant DNA. So the first one, obviously, you have to understand whichever is the gene of your interest, and then you have to isolate that genetic material. Okay. And you know that, okay, where is the genetic material present, students?
Nucleus.
Hmm? But if I talk about prokaryotes, tell me in case of prokaryotes, where is the genetic material present? Hmm? Where is the prokaryotic genetic material present? No one. Does a prokaryote have a nucleus? Yes or no? Am I audible?
Yes, ma'am.
So does the prokaryotic cell have a nucleus? Yes or no?
Yes.
It has a nucleus.
Yeah. Yes.
Everyone agrees.
It's not. It's not like fully formed.
Fully formed. But nucleus is there.
Yeah.
How many of you agree?
Nuclear membrane is not there. I know that.
So if the nuclear membrane is not there, then the nucleus is also not there.
Yeah.
Oh.
See, this is something you people learn in, I think, in class 9th also. See, students, the biggest difference between a prokaryote and a eukaryote is that a prokaryote does not have a nucleus. It does not have any sort of membrane bound cell organ. Instead of a nucleus, there is a thing called as nucleoid that is present in prokaryotes. Eukaryotic organisms, they have a well-defined nucleus, they have well-defined cell organelles and everything. But when we talk about the prokaryotes, they do not have, always remember, they do not have a well-defined nucleus. Nuclear membrane is not there. Nucleus itself is not there. Only a region called as nucleoid, this region. Okay, nucleoid. Now, talking about the process, or you can say the steps of RDT, the first one is isolation of genetic material. Meaning, once you have identified that, okay, this is the gene that I want to use, or this is the gene that I want to send it into the host cell. So, you isolate that genetic material. After that, you cut pieces of it, isn't it? Then only, because that long genetic material is always very long. So you can't use that long of a genetic material all at once. So you make certain cuts. So you cut the DNA at specific locations using the restriction enzymes that are known as, restriction enzymes are also known as
Chemical
something what?
scissors? No, chemical scissors. No.
What are you saying? Chemical
Chemi-
scissors. Yeah, you are right. Not chemical scissors, but molecular scissors. They are scissors only. Okay, so they are molecular scissors. Very good. Now, after the scissors have done their work of cutting, then we separate them. Because imagine, have you ever seen your mother's chopping lady finger? Hmm? Have you people seen, like, if she chops it, all the fragments, all the pieces, they keep getting stuck at one place only, isn't it? It's like getting collected at one place. So you have to separate it to cook it, right? While you're cooking it also, you have to separate it. So once the DNA fragments have been made, then those DNA fragments, they have to be separated and isolated. You have to take out the fragment to use it, right? You can't use all the fragments at once. You have to pinpoint and take out one fragment. After that, you have to amplify the gene of interest. Amplify means make multiple copies of it, and you do so by using PCR. We'll talk about it. Then comes, I think PCR I've taught you, right? Have I taught you PCR before or no? Okay. PCR stands for polymerase chain reaction. So we're going to see how that happens. Then there is ligation of the DNA fragment into a vector. Meaning now we are joining the fragment onto a vector, meaning a carrier. The most common vector is most common vector.
Bacteria.
Correct. In that, which one? Like a plasmid,
Fungi.
isn't it? A plasmid, like this thing, PBR 322, isn't it the most common one? Okay, so we do the ligation. Ligation means joining. And for that, what do we use? What do we use for ligation? If we use molecular scissors for cutting, then what are we used for joining?
Molecular glue.
Molecular glue, which is an enzyme called as DNA ligase. Ligation means joining, attaching. So it is done with the help of an enzyme called as DNA ligase. Now, after that is done, then we do transferring. Now we finally transferring of the recombinant DNA. Transferring of the recombinant DNA. Where are we going to transfer it now? Tell me, use your common sense. I've done all of these things. Now I want to transfer it. So where will I transfer? To me. Then the transferring of the recombinant DNA into the
Vector.
huh?
Vector.
What?
Vector.
Huh?
Vector.
Duster?
Vector. Vector. V.
I can't hear you. Please spell it out. I
He's
can't hear
saying
you.
vector.
Vector. No,
Vector.
vector we have used vector. We are not going to use. Why did he leave the class? He left the class. Yeah, see, no, not the vector.
I
Let him
think he
come back.
thought his internet was
Hmm. We're not going to transfer it into the vector.
into the gene.
Into the gene. Yes, yeah, we're not going to transfer it into a vector, okay? I thought you got angry and you left the class.
No, ma'am.
Okay, see, the vector part is over. Where is the vector part over, students? Why do I feel like none of you understand anything from this topic? I have taught 5 steps to you up until now. Let us see the 5 steps. Do you people have any difficulties in that? Don't just say no just for the sake that I'll continue and the class will get over. Please try to learn. Okay, you have to learn here. So tell me, or have you people gotten laid back because I said I'll do the revision of the chapter? Hmm? Have you gotten laid back? I hope not. Okay, so let's talk about it again, step by step, because you people only have to answer this, the 6th one. I'll not answer. The first step, you recognize the gene of interest. You know that you want to use this particular gene. It can be anything. So you isolate the genetic material to do that because the gene is the genetic material, correct? So you isolated it. You took it out. Secondly, now you want to cut it because you can't use a long genetic material. You can't. DNA is very long. Everyone agrees? DNA is so long. So as to use the DNA, I'm saying DNA because DNA is the preferred genetic material, right? It can be RNA, but most of the work is done on DNA itself. So after you've taken out the DNA, you cut it into pieces. And that is done with the help of restriction enzymes, which we call as molecular scissors. They are scissors. So you snip it, you snip it. After that, if you have taken a long piece of DNA, that means that there will be several fragments, several pieces after you've cut them. So now you want to take one piece or two pieces out of it. So you separate all the pieces together, right? You not together, separate them apart. And then you want to isolate, meaning after you have spread it apart, you took one of it because you want to use that one. Or you took two of them because you want to use two. You can't use all of it at once. Getting my point? Everyone's following me? So then you isolated your desired fragment. Now, for example, in a DNA, in one DNA, okay, there can be many genes. Everyone agrees to this point? In a DNA, there can be multiple genes present. So whatever is the piece of the DNA that you took out, the piece that you isolated after cutting, Now you have to make multiple copies. Now there is a reason why. For example, all of you have done practical in your lab, chemistry lab or chemistry lab mostly.
Yeah.
So do you see that your teacher asks you to take 3 to 4 readings?
Yeah.
Isn't it? Everyone is asked to take
Yes.
several readings. Why?
Um,
You don't know. None of you know why you are asked to take several readings. I'll tell you. Yes, yes, Mariam. About to say something? Just take a guess. Why do they ask you to take
be
several readings?
to be more accurate, like to find the
Hmm.
average.
Not to find the average, but to be more accurate. Because for example, you're doing the experiment for the first time. You did the whole procedure. Maybe there can be human error, right? Because we are humans, we tend to make mistakes. So there can be human errors. So if you have taken 5 different observations, it means that one of them will be correct at the end, or 2 of them might be correct. Not all of them will be wrong, right? So same thing, if we took out one part of the gene and we want to work with that, do you think it like will be perfect and do everything in a perfect manner? It's not possible, right? So what will we do? We'll amplify that gene of interest. Amplification, you people understand how you amplify a sound, isn't it? While someone is playing music, you amplify the sound. Same way, amplification means increasing, making multiple copies in terms of RDT. So you took that gene of interest, now you made multiple copies of it. So now from you started with one part, now you have 100 to work with. Now if you make mistake in 10, then the next 10 is again available for you. You're getting all of them have the same property because you have amplified that one gene of interest, not the whole DNA. This much 4 steps are clear to everyone. You people have understood? Sure.
yes.
Hmm. After that, you took the amplified DNA. Now what do you want to do? You want to put it onto a vector, a carrier. For example, um, you have a business, okay, any kind of business. Some of you are painters, let's just say. Okay, you made a painting. Now to sell it to the person who has ordered it from you, what will you do? You'll pack it up to send it to a courier company, so that that particular courier company would take that order to the customer, right? Same thing we are doing here. Now we need a courier company, and that courier company over here is a vector, because vector is a carrier. Imagine, don't, when I say vector, you think about a vehicle. It'll drive your gene vehicle, right? Any kind of automobile. So now what you do, you take that DNA fragment and you ligate it onto a vector. Ligate it means join it. Because obviously, if you're going to courier out something, you put it in a box, you tape it up, you wrap it up so that your piece does not get damaged. So same thing you're over like doing over here. Like how will the vector carry it for you? You have to put it into the vector, right? You have to stick it onto a vector. So that is what you're doing. You're ligating the DNA fragment onto the vector. Clear? This much is clear? Now
Yes,
coming
ma'am.
on to the next step, that is transferring. Now see, after this is done, when you have taken the gene of interest and you have ligated it onto a vector. So gene of interest plus vector is equal to recombinant DNA. Gene of interest plus vector is equal to recombinant DNA. So now up until the 5th step, what you have made is a recombinant DNA. Clear to you all? I'll write this over here also. That vector plus gene of interest is equal to recombinant DNA. Clear? So after the recombinant DNA has been made, now you have to transfer the recombinant DNA into the
Host.
Host cell.
Host cell. Very good. Exactly. Now this is the customer. You're sending it here. The courier is being sent here. So now this is the customer, which is the host cell. Now you people have understood. Very good. It gave me such a good satisfaction. Clear to you all? Okay. So now we have completed the sixth step. Now, after it has been transferred into the host cell, what do you expect the host cell to do? You can like imagine an answer. You don't have to be right. For example, your courier reached the customer. Now, what do you want the customer to do?
The
Don't say
customer
open the
should
pack.
use the
Don't say open the
product.
pack. The customer should use the product, right? And also write a review about you that, okay, it was good. The product was nice. Maybe post a story, right? All of these things you would want to do. So here the host cell will perform what it is supposed to do. After receiving the recombinant DNA, it will start preparing more of it. It will incorporate it inside itself. Like you want the customer to use it, right? So the host cell will use this DNA now. This DNA, this recombinant DNA will now become the part of host DNA. Just like how your product will become a part of the customer's life, isn't it? If it is a wall hanging, the person will hang it. If it is something usable, so the person will use it. So now the recombinant DNA will become a part of the host DNA. So the next step is culturing the host cell, because we are doing it in the lab. So everything, all of these things are being done by us. It's not happening naturally. It is an artificial process. So now we do culture the host cells. Culture means have you people ever done any experiment with bacteria? No, right? I don't think it's there in school. Okay, see, culturing means, for example, you take a bacteria and you take a petri dish. You people understand what a petri dish is? Right? Petri dish? No? Those round circular dishes that are provided, you also not understand this. Okay, see, you have not seen this because you people are in school. See, there is like a glass dish which is circular. Okay, it is a dish made up of a glass and it has a lid also on top of it like this. So what that dish is used for is that you will put a nutrient medium, meaning you will put, just like how do I explain this? See, a nutrient medium is like a jelly-like substance. It has carbon source, it has food, it has vitamins, minerals, everything that the bacteria will require. So on that plate, you put everything and you put the bacteria here. Now the bacteria will take up the nutrient medium. It will reuse the nutrition and it will reproduce. It will make colonies, meaning it will start increasing in number. So that is culturing. Okay, that is culturing. So once we have transferred the recombinant DNA into the host cell, we culture the host cell in a nutrient medium at a large scale. Meaning we make many copies of host cell now. Why? I'll tell you how. See, nutrient medium at a large scale. I'll show you with the help of a diagram so you will understand it better. And lastly, you will extract or extraction of desired product. Whatever is the product, you will now extract it. And now the process is completed. Now see, I'll show you here. For example, this was the gene of interest. This is a vector. Okay. So now I've added the gene of interest here. What did it make?
Recombinant
Recombinant
DNA.
DNA.
Rest of the students are also understanding, right? Those of you who are not answering in the class, you are understanding it, right? Sure. Don't hesitate to answer. And if you like, I can understand like everyone has a different personality. Some people speak, some people don't speak. That's all right. As long as you are understanding, I don't mind if you speak in the class or not. So for the rest of the students who are not interacting in the class, I hope you are understanding. Huh? Now
Yes,
you can
ma'am.
interact. Huh. Yes. So if at any point you don't understand, please ask. So we took the foreign gene, we added it onto a vector, and we created a recombinant DNA. Now we take this recombinant DNA and we add it onto the host cell, right? Now, for example, this is the recombinant DNA added here, meaning now it becomes a part of the host. It becomes a part of the host. Now you take this host and you take a petri dish and you supply it with a jelly-like substance so that the bacteria could use it to multiply. So if this is the host with the gene of interest. Now when it will multiply, it will create daughter cell, correct? The bacteria multiplies by which kind of division? The division of bacteria is called as?
Mitotic.
Hmm?
Mitotic division.
Yeah, it is a type of mitotic division, meaning you people remember binary fission, right? Binary
Oh
fission, you might,
yeah.
in that what
Divide into
happens? First, the cell divides into two, but before that, the genetic material also separates. So now this cell, which is the daughter cell, will have the recombinant DNA. Another cell, that is the daughter cell, it will also have recombinant. Furthermore, these daughter cells will divide and form the same thing, meaning now the genetic or the gene of interest, whatever was your gene of interest, it is getting multiplied. Are you seeing that? It is being like from 1, it went into 2, then 4, then 8, then so on. And it's not this much, right? So this is known as culturing. Culture, bacteria culture, meaning to increase. Because normally any organism that performs, that has to live, that wants to live, what will it do? It will reproduce, right? So that's what bacteria is doing. And the nutrient medium is a medium wherein you have provided everything suitable to bacteria. For example, if I ask you to do an experiment on a human, okay, on a human, it's not possible, but it's imaginary way to explain these things to you. So you take a box. Hmm? You want to make the human survive in the box. What all things will you put it in the box?
Food, water,
You'll put food, you'll put water. What else?
the opposite gender to reproduce.
Huh?
The opposite gender to reproduce.
Correct. You'll put the opposite gender because the humans would reproduce sexually, obviously. Very good. Are you people forgetting a very important part?
Air.
Air, oxygen. Very good. You'll put oxygen, no? The human will
Yes.
die. Okay. And since human is a social being, so you'll put one or two more people also so that the human can interact. So what are you doing? You are basically creating a very nice surrounding, an environment for the human to dwell. Same thing you're doing for bacteria. You can't do it for a human, but you do it for a bacteria. Now, obviously, bacteria does not require a mate, so you don't have to put that. But bacteria, some bacteria might require oxygen, some don't. Or some bacteria require glucose as a source, right? You put food for humans, so you'll put glucose for bacteria, or you'll put lactose for bacteria. That depends. This much is clear? And once the desired product has been synthesized, you extract it and then you send it into the market. That is what is recombinant DNA, all in all. Are all the steps clear to you?
Ma'am, what is the desired product that we extract?
That depends on you. For example, if you want to create um, um, a toxin that is produced in bacteria, so you take that gene of toxin from the bacteria, you culture it, you make multiple copies, then you add it into the host cell. You want one host cell to make the toxin that the bacteria makes. So you take the gene from bacteria, you put it into the host cell. Now the host cell makes the toxin. So now you have a host cell that makes toxin. That is your product. For example, normal people have insulin produced in their body, right? All
Yes.
of us have insulin. Insulin is a hormone. Which organ secretes insulin?
Pancreas.
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 Pancreas. Very good. Pancreas. So now, if there is any person who does not have a well functioning pancreas, so that person cannot synthesize enough insulin or a good amount of insulin. So now you make the bacteria produce that artificial insulin. So if you are doing all of these steps to make artificial insulin, so that is your product. You extract that and you sell it into the market. Right?
We can
So
sell it as
just
tablets.
hmm,
Artificial insulin.
hmm. There are many things that are developed by recombinant DNA technology. There are many products that these all things you'll have to learn in the next chapter. That is application.
This is interesting.
Yeah, it isn't. That is what I'm saying to you people. It's a very interesting chapter. It's new to you, but it's very interesting. So all of these steps are clear to you all. Does anyone have any doubts in any of the steps? Because now we are going to discuss each of the steps in depth. So no one has any doubts. What about the students that are very less interactive? Okay, one more question, students. Have you ever had the team tell your parents about your performance? How you're performing here?
Yes, ma'am.
You people listen to that?
Yeah.
Your parents tell you that they got to know your performance?
Yes.
So see here, apart from teaching you all, I also evaluate you while you're in the class, the way you behave, the way you interact with me, no, like how many questions you ask, whether you understand or not, right? So all of these things are being evaluated by me, and at the end, they ask me what is the feedback of this particular student. So students, be interactive in the class because I don't think now there's anyone in the class who's shy with me. Is there anyone who's shy? Like who feels shy to speak to me? I don't think I am that way that I cannot make anyone feel shy. I think so. Is there anyone who feels shy to talk to me? Tell me, students. Siddika,
No, ma'am.
Maria, Khadija, Amna, Mariam Lohani. Tell me. You people don't feel shy. You people feel comfortable in like being taught by me.
Yes, ma'am.
Yes.
Okay, so if you do so, then please interact in the
Yes.
class. The more you'll interact, the better you'll learn. Ask from these two. These two Changu Mangus, Mariam and Yahya, you tell me. You interact, you learn better when you interact, right? When you ask lots of questions, when you make faces and I teach you again. That is how you learn better. See? And if you learn it like that, you'll score better. Trust me. If you people might have noticed this one fact that if you ask a question in the class, you are validating your study. Yeah, you're validating what you have studied. And once you are like your question gets answered, you remember it forever. Now, I don't think anyone in the class will ever forget the courier and the customer thing. I relate it with normal life things so that you people can understand. So, though all in all, I always drift from the topic. All in all, I want to say is that interact in the class. Okay, if I ask you to switch on your video, it's not that I want to see you. I mean, I want to see you, but it's not that I want to see you. It's like I want you people to interact. Because if I see your faces, I clearly tell whether you're not understanding or not. I can tell it by your faces. I can read your faces, trust me. So interact in the class. Okay, so you people have understood all of the process of recombinant DNA technology. Is there anything that has a doubt?
No, ma'am.
Okay, good. Now you people can note it down.
Done, ma'am.
Everyone's done?
Done, ma'am.
Done, students?
Yes, ma'am.
Okay, now you can draw this also so as to remember. If you have any difficulty at any point, you can draw this. Done?
Done, ma'am.
Okay. Any doubts, students?
No, ma'am.
Okay, now do one thing. Please open your NCERT books and tell me, are the processes defined in the book? I'll also check it. Just 1 second.
Yes, ma'am. It's explained.
Okay, it's explained.
Yeah.
All right. Okay. Okay, so to begin with, we'll start with how the first step takes place, that is isolation of genetic material. See, for the isolation, first you have to understand whatever is the gene of interest that you want to do so, and then you have to separate the particular part. Now, how do we do so? If we talk about the bacteria, okay, the basic step in the total DNA, like when you want to extract it from a culture, for example. So we're going to talk about the step one. So for example, if this is the vessel that has bacteria present in it, obviously the bacteria will be present in some kind of medium. So here the bacteria is present. Now you want to do centrifugation. So this would undergo centrifugation. Okay, you will agitate it so that settling of things will be done at the bottom. Then you take a test tube, and there you see that after centrifugation, the pellet of cells will be left. Pellet, meaning it will get collected in the bottom. So pellet of cells will be there. Then
What
after
is
that,
centrifugation?
it is a process wherein you, like just like how churning is, meaning constant rotation. You
Oh,
take the thing and you put it under constant rotation.
okay.
And then the pellet of cells, when you take that cell, so you'll do the cell extract. Like this will be the cell extract, meaning you've taken out the pellet from here, pellet of cells, and you added it onto a next test tube. And then later on, finally, after the whole entire process is done, you will have pure DNA in it. So how do we do this actually? I just showed you the whole entire thing with the help of diagram. Now see, first of all, the bacterial culture is grown. Okay, the bacterial culture is grown, and after it has been grown, and then it is harvested. You people understand the meaning of harvested? Harvesting. What is harvesting? No one knows harvesting. Like how you do rainwater harvesting, you do harvesting in the field of agriculture also.
Yeah.
Harvesting means to collect something, right? So after the bacterial culture has been grown, you collect it. Then the cells that are present, obviously it is a bacterial cell, so you don't need the whole cell. For example, if this is the cell, it has genetic material present in it, it has ribosomes present in it, it has vacuoles present in it, it also has some RNA molecules and all of these things present in it. But you don't need this whole apparatus. You just require the DNA. That's all you need. So you have to remove all of the other components to get the pure DNA. So the cells are broken open to release the components, the other components of the cell, the other components of the cell, which are of no use to us, which are of no use to us. And this particular, you can say, removal of the components, it is according to the cell that you are doing. For example, if you have cultured bacterial cell, do you people know that what type of enzyme or what type of chemical can you use to kill the cell of the bacteria?
No, ma'am.
Okay, I'll tell you. What is, okay, you people, do you remember what is the cell wall of bacteria made up of? No? It's made up
Cellulose
of,
and pectin.
that is for plant. Cellulose is for plant. Okay, bacteria has peptidoglycan. Anyway,
I
but
said pectin.
pectin, you said pectin, I said peptidoglycan.
It's different. Oh.
It's very different. See, you can treat the bacterial cell with a with sort of like an enzyme called as lysozyme. Lysozyme. It is an enzyme that can degrades the bacterial cell wall, made up of peptidoglycan. Okay, so it degrades the peptidoglycan. Any doubts? So if you have removed the cell wall, what will you have? If you remove the cell wall of the bacteria, what are you left with? Okay, what is the cell of the fungus made up of?
I don't know.
Chitin. So you use chitinase to degrade that. Also, we'll talk about the plant cell. So the plant cell is made up of cellulose. All of you know that. So the cellulose can be degraded with cellulase enzyme. We'll discuss more about it in the next class. But have you people understood the basic mechanism that you are degrading the cell wall because you don't need every component of the cell.